Hi everyone. This revision lesson will put together what we have learned about the specific immune response. It's just a general overview, so make sure that you do read your textbook in detail. To start with, the initiation of the immune response occurs when a pathogen with a foreign or non-self antigen on its surface enters the body, and this is detected by a macrophage. The macrophage will engulf the pathogen by phagocytosis, and that will destroy it. From here, two things happen. Some of that pathogen antigen is displayed on the surface of the macrophage. These fragments are part of that initial immune response. The pathogen fragments are displayed and the macrophage becomes an antigen presenting cell. The rest of those pathogenic fragments are expelled from the cell through the process of exocytosis. Our antigen presenting cell will now migrate to the lymphoid tissue, as this is where the B and T lymphocytes are located. Here, the antigen presenting cell will stimulate helper T cells to respond to those foreign antigens that it is presenting. The helper T cells, remember, will release chemicals called cytokines. Cytokines activate B and T lymphocytes to initiate the two types of immune response. Those are the humoral and the cell-mediated response. All right, so let's start by reviewing what happens in the humoral response. So that's the cell response to do with B cells. Initially, B cells, when they are stimulated, they will start to enlarge and divide, and that will form a group of cells called a clone. Clone cells are mostly plasma cells, They secrete a specific antibody, so it's the plasma cells that do that. These cells will circulate in the blood, lymph, and extracellular fluid to reach these foreign microorganisms. B cells that do not differentiate remain as memory cells. These memory cells will spread throughout the body, and they will allow a more rapid response should that pathogen invade a second time, and that is the basis of immunological memory in the secondary response. So how do antibodies work? They work by combining with foreign enzymes or bacterial toxins to inactivate them by inhibiting their reaction with cells or other compounds. And they can do this in a number of different ways. They also bind to the surface of viruses, and they prevent it from entering, or they coat a bacteria, and these different mechanisms allow those pathogens to be easily consumed by phagocytosis. So neutralization, agglutination, and antibody binding are the three mechanisms that we need to talk about in regard to enhancing phagocytic action of macrophages. Fourth type of action we need to look at is lysis. This mode of antibody action is designed to perforate the cell membrane, which will induce lysis or rupturing of the cell. So at the same time as the B cell response is taking place, the T cell response is also gearing up to take on pathogens. So again, the helper T cells will stimulate activation of cytotoxic or killer T cells via cytokines. These activated killer T cells will clone themselves in a process known as clonal expansion, similar to B cells, and this will produce much more of them. The cytotoxic T cells will track down infected cells or viruses, that contain viruses, and once they are in contact, they will bind to the foreign antigens presented on the surface of the infected cell and release chemicals called perforins. Perforins will essentially punch holes in the cell membrane, and that induces lysis. At the same time as this action by the cytotoxic T cells is taking place, the infected cell itself will secrete a chemical called lymphotoxin, and that activates the cell's own self-destruct mechanism. Now, both of these actions are designed to prevent the virus from having a host to reproduce in. So if you kill the cell, then it can't cause any further damage. That's the thinking behind that. So during that clonal expansion that we talked about at the start, two other types of cell are produced. So I've only written about one of them here, but just to remind you, memory cells are produced and they remain in the lymphoid tissue, again for that secondary immune response. But we also have suppressor T cells, which will dampen down that immune response when it's no longer required. All right, going back to the actual immune response itself, the immune response will involve a third chemical, and this is the second chemical triggered by the T cells. These are perforins, these are lymphokines. Perforins are the first ones. The lymphokines will stimulate enhanced macrophage activity to migrate towards the infected cell site, and it will destroy the pathogen.